Hey folks, so welcome back to Range Academy. So in the past few sessions, we have been learning what an action potential is. How is this action potential generated? How it is propagating through a neuron? How is this action potential transfer from one neuron to the other neuron? And the most recent session, we did learn that how this information is transferred from a neuron to a muscle, or in simple terms, we just learned neuromuscular transmission. Okay, and in the end of that session, I did promise that we will be learning on how muscles contract with the help of that action potential, and that is what we will be covering in this session. Okay, so as you can see, there are a lot of figures. Okay, so let's just recap the story. Okay, so we suppose a stimulus of enough threshold is applied to a neuron. Okay, and imagine it reaching the neuronal end. Okay, and how did it reach the neuronal end? When a stimulus of enough threshold is applied to a neuron, the mechanosensitive sodium receptors open. Okay, and it leads to a slight influx of sodium so that the threshold level is achieved. Once the threshold level is achieved, what happens? The voltage gated sodium channels, they open up and they cause a large amount of sodium influx, thus leading to the cell being more and more positive, and the cell will eventually try to reach its equilibrium potential. But before it can attain that equilibrium potential, what happens? The inactivation gates of those voltage gated sodium channels close, and thus now due to the action of the leaky potassium channels and the voltage gated potassium channels, which close much slower than the voltage gated sodium channels, potassium starts to leak out. The positivity inside the cell starts to decrease and it starts to become negative, and the cell eventually reaches its resting membrane potential. Okay, and that phase is what we called repolarization of the cell. Right, this was a story. For all those who haven't seen those videos, please do watch it. The link will be provided in the description below. Okay, so suppose our action potential have reached the nerve end. Okay, this is the nerve end. Now, if you recall the last session, what happened here? The action potential reached here, and during the production of action potential, the cell inside of the cell becomes more and more positive. And when the cell potential inside becomes around negative 35 millivolt, what happens? Our voltage gated calcium channels are activated, thereby allowing the influx of large amounts of calcium into the neuronal end. And what does this calcium do? We told that there are large number of vesicles in the nerve ending or terminal end which contain acetylcholine, and these vesicles are specialized structures that they also have certain receptors or proteins, and the nerve end also has certain proteins. In order for this acetylcholine to be released out, what should happen? These two proteins should bind to each other, and that is mediated by the calcium that just got inside. So calcium mediates the binding of these proteins, and what were these proteins called? The V snare proteins and the T snare proteins, right? And this binding allows the exocytosis or the blending of the vesicle with the neuronal membrane, eventually leading to the release of acetylcholine into the gap between the muscle and the nerve. And what was this gap called? The synaptic cleft, right? Now, what does acetylcholine do? Acetylcholine binds to the ligand gated ion channel, or more specifically the nicotinic receptors, which needs acetylcholine to activate itself. So acetylcholine binds to this nicotinic or ligand gated ion channel and activates the channel. And when these channels are activated, what happens? There is an influx of sodium into the muscle cell, right? And when sodium influx occurs, what happens? The cell starts to become more positive, or in other terms, it starts to depolarize, right? And as the cell starts to depolarize, the membrane potential becomes more and more positive, and what happens when it reaches the threshold level? Okay, when the membrane potential reaches the threshold level, which is the voltage required for the activation of our voltage gated sodium channels that are present on the nearby side. Okay, when the cell membrane potential reaches the threshold potential, there is furthermore influx of sodium and thus leading to the membrane potential generating being an action potential. Okay, now what was this? The resting membrane potential being the threshold potential in a muscle cell. This is what we call the end plate potential. Okay, or the potential that is being more and more positive so that the membrane cell achieves its threshold potential. That is referred to as the end plate potential. Okay, and eventually the story continues. The inactivation gates close faster, and the cell starts to lose potassium via voltage gated potassium channels and leaky potassium channels, and the cell starts to become more and more negative. That is, the cell starts to repolarize, and then it hyperpolarizes, and then after the voltage gated potassium channels close completely, the cell attains its resting membrane potential. Okay, this is what happened, right? Now, when the action potential is traversing through the muscle cell, what happens? The action potential eventually goes down through this loop. Do you see a loop here? Okay, this is the plasma membrane of the muscle cell. Okay, the plasma membrane forms certain folds into the cell. Okay, and these folds are called the T-tubule. Okay, or this T stands for transverse tubule. Okay, transverse tubule. And when the action potential runs through this T-tubule, there are certain activities that occur here. Before that, do you see box-like structures present on either side of the T-tubule? These are the endoplasmic reticulum that are present in muscle cells. Endoplasmic reticulum present in the muscle cells are referred to another term, and it is referred to as the sarcoplasmic reticulum. Okay, sarcoplasmic reticulum. And the sarcoplasmic reticulum do have a speciality. They are rich in calcium. Okay, they are rich in calcium. And why? Because they have the presence of a protein called calsequestrin, which are calcium concentrating proteins present exclusively in muscular cells or in skeletal cells. Now, this endoplasmic reticulum or this sarcoplasmic reticulum, this T-tubule and this sarcoplasmic reticulum, these three are referred to as a triad. Okay, this is referred to as a triad because there are three elements: two sarcoplasmic reticulum and a T-tubule. And what is a T-tubule? It is the invagination of the plasma membrane of the muscle cell. Simple. Okay. Now, the action potential runs through the T-tubule. What happens? You can see there are two kinds of proteins I have drawn. One on the sarcoplasmic side, one on the tubular side. Right. These proteins that are present on the T-tubules are referred to as the dihydropyridine receptors, or in simple terms they can be called DHP receptors. Okay, and these receptors that are present over the sarcoplasmic reticulum are referred to as ryanidine receptors. Ryanidine receptors. Okay, when action potential runs through this T-tubule, it causes the binding of this dihydropyridine receptor with the ryanidine receptors. They bind each other, and what happens? The DHP receptor or dihydropyridine receptor pulls out the ryanidine receptors. So what happens? When this receptor is pulled out from the sarcoplasmic reticulum, there forms a gap. Right? There forms a gap in the sarcoplasmic reticulum, and through this gap, who escapes? Calcium escapes. Okay, calcium escapes from this gap. Now, I think you can recall this structure which I have drawn here, right? This structure is the sarcomere. We learned detail in the last session. Okay, so this calcium escapes and we have a sarcomere here. So what happens next? Okay, so on the board you can see too many figures, right? We'll go one by one. Okay, so I think you're familiar with this figure about which we had discussed in the last session, and it was called the sarcomere. And what does it consist of? We have a Z disc or the Z line. Okay, we have the thick filament, also called the myosin. We have the thin filament, that is called the actin. And now we have certain anchoring proteins too. The anchoring protein that binds this thick filament to the Z disc. The ends of the thick filament to the Z disc is referred to as titin. Right. Now, I'm zooming this thin filament. Okay, so you can get a better understanding. We had a helical protein running through this, right? See, we have a helical protein running through this. Helical globular protein. And what was it called? It was called the actin, right? The monomer of actin was the G-actin, that is globular actin. When I take one piece of this or one ball, it is called the G-actin. When I use this G-actin to form a small chain, it is called the F-actin. And when this F-actin together form a group, they are referred to as the actin. So this overall is the actin filament. You can also see a thread-like structure running through this, right? See, this one. And what was it called? It was called the tropomyosin. And what did it do? It made sure that the active sites of actin filament are closed or masked, right? And what was this protein? This was our troponin complex, right? Why was it called the complex? Because it had three binding sites. One was for calcium. Yes, you are right. One was for calcium. One is to the actin filament and the other one was to the tropomyosin. So one was called the calcium binding site, the other was called the T-troponin, and the other one is called the C-troponin. And the area which binds to the actin filament was referred to as not a troponin but simply the actin binding region. Okay, so we had these three sites, so it is referred to as our troponin complex. Okay, now I have simplified this actin filament to this figure where I just show the actin filament. Okay, active filament here with these are the active sites. Okay, this crescent shaped are called the active sites. And what is this line or this protein that is masking the active site? That is the tropomyosin. And what is this protein called? Our troponin complex, troponin protein complex, right? So I have simplified this actin filament or thin filament into this figure. Okay, I think it is clear now. And who's this? This is our thick filament, also called the myosin. And the myosin also has filamentous structure extending at the ends. And these are the regions that are supposed to bind to two areas. See, there are two heads here, right? One will bind to the active binding site of the thin filament. The other one will bind to whom? The other one will bind to ATP. Okay, so earlier we stopped at the release of calcium, right? And where did this calcium bind? This calcium binds to the calcium binding region of the troponin. Okay, and when calcium binds to the troponin, what happens? It is activated and it pulls out the tropomyosin, revealing the active site. When calcium binds to the troponin protein, what happens? It pulls out the tropomyosin and opens out the active sites. And when the active site is opened, who is there to bind to it? We have our myosin head, one of the myosin head. that is ready to bind to our active site. Okay, but the problem is binding to an active site and causing muscle contraction. It is not something that doesn't require energy. It requires energy. And for the purpose of energy, it also had another site which had to bind to ATP. Okay, now take a close look at me. Consider this to be my thin filament or actin filament with the active sites open, and consider this to be my myosin filament or myosin head. Okay, now what happens when the muscle is about to contract and ATP binds to the ATP binding site of myosin head. Okay, and when it binds to the myosin head, what happens? ATP is phosphorylated into ADP and an inorganic phosphate. That is, ATP changes to ADP and an inorganic phosphate, and the energy released during this reaction is used by the myosin head to do two things. One is the myosin filament is like this. See, you can see the myosin filament is always like almost like in the central region, and the actin filaments are on the either side. Okay, so the myosin filament has to get into position before it can bind to the active site. So that is what is gonna happen. This is the active filament, this is the myosin head. Once ATP is phosphorylated into ADP and inorganic phosphate, it gets into its position. Okay, and then it binds to the active site of actin. So these are the two processes that happen when ATP is phosphorylated into ADP and inorganic phosphate. Now, after this binding, the ADP and inorganic phosphate is released out or it is hydrolyzed. And during this hydrolysis, what happens? The myosin head pulls the actin filament towards the M line. Okay, towards the midline. And now, after this, the myosin head must detach from this existing actin filament in order to bind to another active filament. And for that, who is also needed? Another ATP molecule is needed so that the myosin head can detach, get into position and bind to the another actin filament, and the process continues. Okay, so in order for myosin head to bind to actin filament, an ATP molecule is required. An ATP molecule is also required when the myosin head has to detach from the existing actin filament, get into position and bind to another. So this is the process that is happening. Okay, so we have ATP binds to the ATP binding site of myosin head. It is phosphorylated into ADP and inorganic phosphate. The energy is used to get back into position and then bind to the active site. Okay, and this ADP and inorganic phosphate produced are hydrolyzed or they are released out. And during that process, what happens? This actin filament is pulled towards the M line or towards the center. Okay, and after that, for the myosin head to detach, another ATP molecule must arrive. It should hydrolyze, and thus it detaches, gets back into position and binds to another. This thing, now what you learned is referred to as the sliding filament theory, because the myosin head binds and then it is pulling it towards the M line. So this process is referred to as sliding. So this is the sliding filament theory that explains the contraction of myofilaments, thereby causing muscle contraction. Okay, so let us summarize the sliding filament theory, right? What happens first? The action potential runs through the T-tubule or the triad and it causes a release of calcium. Okay, and what does happen to this calcium? Calcium binds to the C-troponin of troponin protein complex, right? And when it binds, what happens? It pulls out the tropomyosin. Okay, tropomyosin is pulled out, and when tropomyosin is pulled out, what happens? Active sites are unmasked, right? Now what happens? ATP molecule binds to one of the myosin head, right? And it is hydrolyzed into ADP and inorganic phosphate, and this energy released during the phosphorylation is used by the other myosin head to bind to our active site of actin filament or thin filament, right? And this process of binding of the myosin head to the active site of actin filament is referred to as cross bridge formation. Okay. Now, after cross bridge formation, what happens? The produced ADP and inorganic phosphate are hydrolyzed. Okay. And when they are hydrolyzed, what happens? The actin myosin complex is pulled towards the M line. Okay. Now, in order for the actin myosin complex to break and bind to another and for another actin myosin complex, what should happen? Another ATP arrives, hydrolysis to ADP and inorganic phosphate, helps in breaking the existing actin myosin bond. Myosin moves into its position and attaches to another active site of actin filament to form the another actin myosin complex. Okay, and this way the muscle contraction occurs. This is the sliding filament theory. Okay, and remember the actin filaments are always pulled towards the M line. This is the M line, right? It is pulled towards the M line. Okay, now let us discuss on a clinical aspect related to this. When a person dies, what happens? The existing ATP in his body is used for the muscular contraction, right? So, using the existing ATP, new actin myosin bonds are formed, new actin myosin complexes are formed, and muscle contraction happens. Okay, but after the death of a person and after the using of the remaining ATP, there is no scope for the production of yet another ATP. Okay, so what happens? Active myosin complex formed is existing and the muscle is staying in a contracted state, but there is no another ATP available so that the actin myosin bond can break and the muscle can relax, right? And thus you can see that in people who are dead for around within 4 to 6 hours their body becomes stiff. Okay, and that condition where the body is stiff after death is referred to as rigor mortis. Okay, rigor mortis. And what is the physiology behind this? It is because after the body uses the available ATP to form cross bridges, there is no another ATP available for breaking the bond and causing muscle relaxation. Okay, this is one clinical aspect related to our muscle contraction. We are moving towards the end of the session. Now let us discuss certain terms related to muscle contraction. Okay, so this is the thick filament, this is the thin filament. Do you remember what the A band was? A band was something that extended from one end of the thick filament to the other end of the thick filament. Or in other words, the thick filament was itself called the A band, right? So the A band extends from one end of the thick filament to the other end of the thick filament. Okay, so this is the A band. Okay, and what was the I band? I band was something that extends from one adjacent ends of the myosin filaments. So I band here extends from this point to the nearby myosin filament. Okay, from this point to this point. So this is the I band. Okay, now we have a new term, the distance between adjacent thin filaments. Okay, earlier we discussed that the distance between adjacent thick filaments was called the I band. The distance between the adjacent actin filaments or the thin filaments is referred to as the H zone. Okay, So, what happens when the myofilaments undergo contraction? Okay, as told earlier, during contraction, the myosin filaments bind to the actin filaments and pull them towards the M line or the midline, right? So the thin filaments are pulled towards the midline, right? So what happens when the thin filaments are pulled towards the midline? The active filaments come closer, okay, and in majority of the cases they go more than that. They overlap each other, okay. So what happens? The H zone disappears or it decreases during muscle contraction, okay. Now during the contraction of the myofilaments, active filaments come towards each other. And what happens here? The active filaments are also bound to the Z line. So they are also pulling the Z line towards the midline, right? So this distance, right? This distance starts also to decrease, right? And what was this distance called? The I band. So I band also decreases when the muscle contracts. Okay. So now what happens to the A band? When the muscle undergoes contraction or the myofilaments undergo contraction, the A band is angled to its position, right? It doesn't undergo any shortening or lengthening. It stays the same, right? So during muscle contraction, it is the actin filaments that are coming close to each other, which also pulls the Z line towards each other. And thus the I band decreases, the H zone decreases, but the A band remains the same. Okay, so during muscular contraction, the A band remains the same. Okay, so this is a frequent question or favorite question of many of the examiners, where they ask what are the changes that happen in a normal sarcomere and a sarcomere that undergoes contraction. Okay, these are the changes. Okay, so that was all about muscle contraction. Hope everything made sense and everyone did enjoy it. Do show your support and love by subscribing to my channel and liking to my videos. If you have any queries or doubts, please do comment it in the comment box below. I'll be replying to those. Okay. Thank you and have a nice day. Cheers.